सो आज हम पढ़ेंगे एनाटॉमी ऑफ फ्लावरिंग प्लांट्स। Welcome to chapter 6 of plant biology, where we'll explore the fascinating internal world of flowering plants. Just as you can observe structural similarities and differences in the external features of living organisms, the internal structures of plants reveal remarkable patterns of organization and adaptation. This comprehensive study of plant anatomy will take you on a journey through the microscopic architecture that enables plants to survive, grow, and thrive in diverse environments. Plant anatomy is the scientific study of the internal structure and organization of plants. From the basic cellular level to complex tissue level system, will discover how plants are masterfully engineered biological machines. You will learn how cells organize into tissues from organs, and different organs show unique internal adaptation that reflects their specific functions and environmental challenges. Understanding plant tissue systems. Plants are organized into three fundamental tissue systems, each with distinct roles and characteristics. Think of these systems as the plant's equivalent to human organ systems, specialized group of tissues working together to perform essential life functions. Epidermal tissue system forms the protective outer covering of the entire plant body. This system includes specialized structure like stomata for gas exchange and various appendages such as trichomes and root hairs. The epidermis acts as the plant's first line of defense against the environmental stresses, while facilitating essential exchanges with the surrounding environment. Ground tissue system comprises bulk of plant body, performs multiple functions including photosynthesis, storage, and support. These system consists of three main types: parenchyma, metabolically active cells; collenchyma, flexible support; and sclerenchyma, rigid support. The ground tissue fills spaces between epidermal and vascular systems. Vascular tissue system, that plant's transportation network consisting of xylem, water and mineral transport, and phloem, food transport. These complex tissues form vascular bundles that extend throughout the plant, connecting roots and stems and leaves in an integrated transport system essential for plant survival and growth. The epidermal tissue system. The epidermal tissue system serves as the plant's interface with its environment, much like skin in animals. This outermost protective barrier is remarkably sophisticated, featuring specialized structures that regulate water loss, gas exchange, and environmental interactions. As you can see over here, this is of dicot, bean shaped guard cells, and this is of monocot, that is dumbbell shaped guard cells, stomata. These are the chlorophyll, these are guard cells, these are epidermal cells, these are this green, dark green color cells are subsidiary cells, and this is the stomatal pore in which the gases exchange, water pass through, and subsidiary transportation and transpiration occurs. Structure and components. The epidermis typically consists of a single layer of elongated, compactly arranged parenchymatous cells. These cells contain minimally cytoplasm lining the cell walls and the large central vacuoles. The outer surface is often coated with a waxy cuticle that prevents water loss, though this protective layer is absent in roots where water absorption is prioritized. Stomata are remarkable microscopic structures embedded in the leaf epidermis. This question comes that where is stomata lies? So it must be leaf epidermis. This also question came in NEET, that which layer prevents water loss. So it is the waxy cuticle layer. Now each stoma consists of two specialized guard cells that surround a stomatal pore. These bean-shaped cells or dumbbell-shaped in grasses contain chloroplast and have uniquely thickened inner walls that enable them to change shape and control pore opening. The stomatal apparatus include the guard cells, stomatal pore, and surrounding subsidiary cells that work together to regulate transpiration and gas exchange, critically process for plant survival. Now, epidermal appendages. Root hairs are unicellular extensions that dramatically increase surface area for water and mineral absorption. Trichomes on stems and leaves are typically multicellular structures that can be branched or unbranched, soft and or rigid, sometimes secretory. These appendages serve multiple functions, including reducing water loss, protecting against herbivores, and reflecting excessive light. The main function of trichome is to reduce water loss. Ground and vascular tissue systems. The ground tissue system forms the structural foundation of plants, while the vascular system serves as the plant's circulatory network. These systems work in perfect coordination to support plant life and function. Ground tissue system. All tissue except epidermis and vascular bundles constitute the ground tissue. This system includes parenchyma, found in cortex, pericycle, pith, and medullary rays of stems and roots. Collenchyma provides flexible mechanical support. Sclerenchyma offers rigid structural support. In leaves, ground tissue forms the mesophyll, specialized chloroplast containing cells that conduct photosynthesis. Now, vascular tissue system. The vascular system consists of complex tissues forming vascular bundles. Open bundles found in dicots contain cambium for secondary growth. Closed bundles found in monocots lack cambium. Radial arrangement, xylem and phloem alternate along different radii of roots. Then only in roots is a radial arrangement is found, both in xylem and phloem. Conjoint arrangement, it is found in stems and leaves, xylem and phloem together along same radius. As you can see, this is a radial arrangement. This is conjoint and close. and this is conjoint and open. Now, dicotyledonous root structure. So here is the structure of a dicotyledonous root, the transverse root structure. As you can see, here is the root hair, below it is epidermis, below it is cortex, below it is endodermis, below it is pericycle. These are pericycle that connects the xylem and phloem. This is the protoxylem, this is the metaxylem. Afterwards you will know how to recognize protoxylem and metaxylem. The central part is called the pith and the phloem is over here. The dicotyledonous root represents a masterpiece of biological engineering, with each tissue layer serving specific functions in water and minerals absorption, storage, and transport. Understanding root anatomy helps explain how plants anchor themselves and obtain essential nutrients from soil. Epiblemma, outermost layer. The protective outer layer featuring numerous root hairs, unicellular projections that dramatically increase surface area for absorption. These microscopic extensions can increase absorption area by up to 1000 times, making them crucial for plant survival. Cortex. Multiple layer of thin-walled parenchyma cells with intercellular spaces allow for air circulation and temporary storage. This tissue serves as a buffer zone and provides structural support while allowing gases to move freely. This is a buffer zone that the minerals and the water passes through the root hair towards the transport cells, that is the xylem and the phloem. Buffer zone and provides structural support which allows gases to move freely. Endodermis. A single layer of barrel-shaped cells without intercellular spaces, featuring Casparian strips, waxy, water-impermeable bands that control water and solute movement into vascular system. The Casparian strip, featuring Casparian strip, is what? Waxy and water-impermeable bands. What is its use? It used to direction the water incoming and the solute movement towards the vascular system. Pericycle. Pericycle, thick-walled parenchymatous cells where lateral roots and vascular cambium initiate during secondary growth. This tissue acts as the source of new root branches and contributes to root expansion. Central still. This word was very important in the previous NEET papers. Frequent questions were asked, and in the CBSE paper also. Contains vascular bundles, usually 2 to 4 xylem and phloem patches, conjunctive tissues, and small pith. The stele forms the plant's central transport hub for water, minerals, and nutrients. Anything under the endodermis is stele. if this is endodermis, anything inside it is still. That covers the xylem, phloem bundle, vascular bundles, conjunctive tissues. Conjunctive tissues means conjuncting the adjacent xylem or phloem, and the small pith. The still forms the plant's central transport hub for water, minerals, and nutrients. Okay, so inside this, anything is still. Monocotyledonous root and stem structures. Now, in dicot roots, you can see that there were 2 to 4 xylem and phloem patches. This is very, very important. 2 to 4 xylem and phloem patches. And in monocot, more than 6 bundles, xylem bundles are found. That is, therefore they are known as polyarch condition. And here it is known as tetrarch condition. Now here, monocotyledonous plants show distinctive anatomical differences from dicots, reflecting their unique evolutionary adaptation and growth patterns. These structural variations have important implications for plant functions and agricultural applications. Monocot root features similar basic organization to dicot roots, but with key differences. Polyarch condition, more than 6 xylem bundles compared to 2 to 4 in dicots. Large and well developed pith. Central tissue is prominent, no secondary growth. Roots remain primary throughout life. Same tissue layers: epidermis, cortex, endodermis, pericycle, and stele. In monocot stem characteristics, monocot stems show remarkable structure adaptation, scattered vascular bundles, not arranged in rings like dicots, as you can see over here, like this. Sclerenchymatous bundle sheaths, each bundle surrounded by supporting tissue. Closed vascular bundle, no cambium, no secondary growth, like here, as you can see. No cambium is found here, therefore closed and conjoined, there is no cambium, whereas here it is cambium, therefore it is open. Water containing cavities within bundles for efficient transport. Peripheral bundles are typically smaller than central ones, creating an efficient transport network. So, this is very, very important. The increased number of xylem bundles provides enhanced water transport capacity, which is particularly important for monocots like grasses that often grow in water limited environment. This is for monocot roots. Here is the diagram for dicot root. As you can see, it is forming like a ring-like structure. Ring-like structure. Here is the protoxylem. Here is the metaxylem. Metaxylem is larger and protoxylem is smaller. Protoxylem is towards the inner side in stem and metaxylem is towards the outer side. But in roots, as you can see, the metaxylem is towards the inner side and protoxylem is towards the outer side. You can learn like this. In roots, proto is towards the medullary and metaxylem it's towards the proto, and here in the monocot roots, protoxylem it's towards the proto, like primitive, originating, and metaxylem it's towards the medullary. Yeah, that's it. And phloem is outside the xylem. So here it is the xylem, and it is the phloem. Dicotyledonous stem architecture. The dicotyledonous stem represents a sophisticated structural system designed for support, transport, and growth. Its organized tissue arrangement enables both primary growth, elongation, and secondary growth, thickening, allowing dicot plants to develop into large trees and woody structures. Epidermis and cortex. Outer protective epidermis with cuticle trichomes and stomata. The cortex has three distinctive zones: outer hypodermis, collenchymatous cells for strength, middle cortical layers, parenchymatous cells with intercellular spaces for gaseous exchange, and inner endodermis, starch sheath rich in starch grains. This is important. The starch sheath are of which shape? It is like barrel shape. This is important, comes in neat and is conceptual. Pericycle and vascular factual. Pericycle and vascular ring. Pericycle appears as semilunar patches. This is very important points of sclerenchyma above phloem region. Vascular bundles are arranged in a characteristic ring formation. A defining feature of dicot stems, each bundle is conjoint, open, and show enlarged protoxylem development. Medullary rays and pith. Radially placed parenchymatous cells from medullary rays between vascular bundles, facilitating lateral transport. The central pith consists of large, rounded parenchymatous cells with intercellular spaces serving storage and sometimes structural support functions. This is also important. This organized arrangement enables dicot stems to undergo secondary growth through cambial activity, allowing plants to increase in girth and develop woody tissues over time. The ring arrangement of vascular bundles is particularly efficient for supporting the plant's increasing size and weight as it matures. Leaf anatomy of dicot versus monocot. Leaves are primary photosynthetic organs of plants, and their internal anatomy reflects sophisticated adaptations for light capture, gas exchange, and efficient photosynthesis. The structural differences between dicot and monocot leaves reveal evolutionary strategies for maximizing photosynthetic efficiency. Dorsiventral or dicot leaf shows distinct structural asymmetry. Adaxial epidermis, upper surface fewer or no stomata. Abaxial epidermis, lower surface, more stomata. Palisade parenchyma, elongated cells arranged vertically for maximum light capture. Spongy cells loosely arranged with air spaces are for gaseous exchange. Reticulate venation with varying vascular bundle sizes is found in dicot leaves. The differentiated mesophyll structure optimizes both light capture and gas exchange. Okay, from here, this is of dicot. Leaf, and this is of monocot leaf. In dicot leaf, you can see that the adaxial or the upper surface is of palisade mesophyll. Palisade mesophyll due to elongated cells arranged vertically for maximum light capture, and the abaxial epidermis, it is covered with spongy mesophyll for gaseous and water transport. Isobilateral monocot leaf shows structural symmetry and specialized features. Stomata on both surfaces. Equal distribution for enhanced gas exchange. Undifferentiated mesophyll. No distinct palisade and spongy layers. Bulliform cells. large empty cells that control leaf curling during water stress, parallel venation with similar sized vascular bundles, bundle sheath cells around vascular tissue. The bulliform cells act as a natural actuators, curling leaf inwards during drought to minimize water loss, and it is mostly found in grasses because they are monocot. This is important. These anatomical differences reflect each group's evolutionary adaptations to their respective environments and growth habits. Dicot leaves are optimized for broad leaved photosynthesis, while monocot leaves are adapted for narrow leaved efficiency and water conservation. Yeah, so here are the functions. So now, key concepts and application. Understanding plant anatomy is fundamental to numerous fields including agriculture, forestry, medicine, and biotechnology. The structural principles you have learned explain how plant functions and adapt to their environment, providing insights essential for plant breeding, crop improvement, and conservation efforts. Agricultural application, knowing of tissue system helps farmer understand water uptake, nutrient transport, and disease progression. Root anatomy explains fertilizer placement strategies, while stem anatomy guides pruning decisions and grafting techniques. Pruning means to cut something off from their existence. So while in tea plantation in the hilltops, there are several pruning techniques that are applied to the plants for efficient growth of tea leaves. Plant identification. Anatomical features serve as reliable identification markers. Vascular bundle arrangement, like vascular bundle, endarch, exarch, is it root, is it stems or leaf, then is it conjoint, is it a radial, is it closed, is it open, is it monocot, is it dicot, stomatal patterns and tissue organization, like in guard cells, the bean shaped cells are dicots and the dumbbell like cells are monocots. Tissue organization help botanists distinguish between species and understand evolutionary relationships. Pharmaceutical research. Plant anatomy guides the extraction of medicinal compounds. Understanding tissue types helps locate where specific chemicals are produced and stored, improving drug discovery and production processes. Environmental science. Anatomical adaptation reveal how plants respond to environmental stresses like drought, pollution, or climate change. This knowledge is crucial for conservation and ecosystem management strategies. Remember, plant anatomy is not just about memorizing structures, it's about understanding how form follows functions in the plant kingdom. Each anatomical feature represents millions of years of evolutionary optimization for survival and reproduction. So, now here are some questions. Review questions and further study. Test your understanding of plant anatomy with these comprehensive questions. These exercises will help reinforce your knowledge and prepare you for more advanced studies in plant biology. Fundamental concepts. Question 1 to 3. Compare and contrast the three tissue systems in flowering plants. How do their structure relate to the functions? The three tissue systems, the parenchymatous, the collenchymatous, the sclerenchymatous. Parenchymatous are thin and mostly oval shaped. They are used for multiplication, division of cells, and cholenchymatase is for maintaining the food, for photosynthesis, for mechanical stress bearing. Then for sclerenchymatase, there are mostly dead cells and contains lignin also. In suberin cells. In the sclerenchymatase. Cells. For protection of plants. Explain the significance of Casparian strips in the endodermis. How do they control water and solute movement? As they are as they are made up of sclerenchymatase cells, they are impermeable, they contain lignin, and therefore they have a waxy type layer, suberin. Suberin cells, they have suberin. So, because of suberin, the water doesn't pass through them but pass parallelly to them, and this controls the direction of this water and the solid movement. Describe the differences between open and closed vascular bundles. In open vascular bundle, the vascular bundles are contains cambium in between them, phloem and xylem. Because of this, they can grow radially, increasing their girth and increasing their cells. And it is mostly found in the dicots, and in the closed vascular bundles, there is no cambium and they cannot grow radially, just like coconut tree. They are monocots. Why is this distinction important for plant growth? This is important for and different plants have different requirements. Like monocots have to make travel the water and minerals to higher distances, and dicots have to produce different nutritional things in them. Draw and label the key differences between monocot and dicot root cross sections. Explain the functional significance of each difference. Draw and label key difference between monocot and dicot roots cross section. So in root, as I said, that the protoxylem and metaxylem differences. Protoxylem is towards the medullary and metaxylem towards the proto, that is the inner part. So here in the monocot root, there are more than 6 vascular bundles, that is they are polyarch, and in dicot root there are less than 6, that is 2 to 4, mostly tetrarch. In dicot roots vascular bundles are there. and explain the functional significance of each differences. So in the monocot roots, there are polyarch because they don't have any cambium, and in dicot roots they have cambium, so they can do secondary growth, and because of secondary growth they can grow vascular bundles, therefore they have less number of vascular bundles, tetrarch. How does the arrangement of vascular bundles differ between dicot and monocot stems? As I no, it is the other question, it is for. So like this only you can complete all the questions, all the 10 questions through the slides before I have study you. Practice drawing cross section from memory and labeling all structure. Create concept maps connecting structure to function for each tissue system. So that's the study tip for today's video, and keep studying, and for all the students who are facing issues in their studies, keep grinding hard, because this is just the beginning, and the journey will smoothen out and in no time. So just practice MCQs as much as you can. For any recommendation you can comment on the video.